Yeah, hi everybody. Great pleasure to be here today. I would be in San Francisco this week regardless. I have been for the last dozen or so years because of the JP Morgan Healthcare Conference. So I think it's appropriate that the topic I'm going to address today is healthcare, in specific, addressing the needs for cancer therapy as a result of, although we've made great progress in the cancer field over the last number of years with the recent moonshot and the original war on cancer, there's still a significant number of patients who unfortunately are not benefiting from the therapies that are currently available from a cancer treatment perspective. And so as we talk about this, really, when you look at the emergence of immune-related therapies in the cancer field in particular, we now find that over 50% of cancer patients in the US in particular are treated with immune-based therapies. And of that 50% of the entire cancer population in this country, about 75 to 85% of those patients do not adequately benefit from therapy, meaning they don't get long-term durable responses, which in a sort of colloquial way we talk about as being cures. And because we don't see that benefit for those patients, there's clearly a need for improvements in therapy or additional therapies that address the reasons why patients don't benefit from the current therapy. So if you think about that, you can see on the right-hand side of this slide, there's a graph that really shows the original benefit for patients as a result of chemotherapy was decreased sort of disease burden over time. That was improved with the emergence of precision therapies where we were able to identify which patients might benefit from which drugs. And then with the advent of immunotherapy, we saw that, in fact, we could cure cancers, but only in about 20% of patients. So what I'm going to talk about to you today is understanding why the other 80% don't respond and understanding what therapies will turn those non-responsive patients into patients who, in fact, do really benefit from therapy. So what we're doing at Bectas is really taking advantage of starting right from patients. So the patients who have the cancer are the best place to learn why they don't respond. And so we've taken a unique approach to understanding and gaining insights from cancer patients themselves to define precision solutions that mean we can bring the right drug to the right patients. And we're focusing this on immuno-oncology related mechanisms. There's two really important advantages for this. One is very directly to the patients. The other is to those people who fund the companies that actually bring these therapies forward. But that indirectly also benefits the patients. And so what we really think about is by being precise in the approach that we take, we can bring better therapies to patients faster. So there's an improved success rate in bringing these therapies forward. And the second thing is because of that precision and that improved success rate, what we can do is we can be more capital efficient in the way in which we develop the drugs. And so we can create value from an investor standpoint and value from a patient standpoint in demonstrating the particular drugs work. Now, in a large ecosystem of biotech companies, that also means we can quickly identify those things that don't work, set those aside, and refocus capital that would be really sort of sunk cost into unsuccessful ventures, and that can be repositioned into others. I'm going to, of course, make the argument that Bectas is primed for success because we're taking this approach, and that capital efficiency combined with that increased success in drug development leads to us to improved therapies for patients. So how do we do that? Well, we take, as shown in the Venn diagram here, we take unique insights from human patients. We combine that with, and that leads us to both understanding what targets we should make drugs against and how to find the patients to deliver those drugs to. We combine that insight from human patients with a deep understanding of the biology of the particular targets that allows us to design the right drug for that particular target. And we use a blood-based system to identify those patients who will benefit from each drug. And that's a really easy thing that can be translated into clinical settings in community hospitals through to large academic cancer centers. And so it really improves the breadth of patients that we can address. And so taking these approaches at Bectas, it's led us to identify myeloid-based suppression of the immune system. So suppression of the immune system through a particular cell population as being the driver of half of the non-response to immunotherapies today. And that, when you look at that from a patient standpoint, that's over 400,000 patients per year in the US alone. You're looking at a multi-billion-dollar market per year from a commercial standpoint. And Bectas has developed 3 monoclonal antibody programs that target different myeloid-based suppressive signals. and the biomarkers or companion diagnostics that will help us identify the patients who will benefit from each of those therapies. The depth of biology is significantly contributed to by the work of one of our scientific advisors, Dr. Jim Allison, who's out of MD Anderson Cancer Center. Many of you may know him. He won the Nobel Prize in 2018 for his work specifically in immuno-oncology, and he has a particular interest since that time focused on myeloid biology because of its importance in conferring resistance to existing immune therapies. So the team, we've built a really good team. In addition to Jim as a scientific advisor, I'm the president and CEO and co-founder of the company. I have over 25 years of experience in biotech and pharma. I brought 25 drugs into clinical development, including 4 approved drugs that treat over 1 million patients per year, earning a revenue per year of about $30 billion across those 4 approved oncology drugs. So a reasonable amount of experience on my side in terms of operational aspects. Dr. Keith Flaherty out of Mass General Hospital leads clinical and translational research there in the oncology space. He's a world-leading expert in the concept of precision therapies for oncology, has founded multiple companies that have been successful both in bringing drugs to market and to patients, as well as from a business standpoint. Dr. Linda Chin, who's out of MD Anderson originally as well, who's a founding board member, as one of the CEO of one of our partners, Apricity Health, and Jim, of course, on the scientific advisory board as well. So the concept of precision then is to say that rather than taking a drug and treating all of the multicolored cancers that you see on the left-hand side of this slide, we identify which patients fall into which color. So who are the green cancer patients? And let's deliver a drug that addresses green cancers to the green cancer patients and not a drug that addresses blue cancers, because that won't benefit patients with green cancer. So this is the concept of precision, and that's very, very simple and straightforward. But the advantages here are that when you treat patients with green cancer with a drug directed to green cancer, you obviously are going to see a much higher success rate in terms of benefit to those patients. And because you don't treat those patients with a drug addressing blue cancers, you're not going to see any toxicity associated with the blue cancer drug in green cancer patients. So you benefit patients both in terms of increased efficacy and in terms of decreased toxicity. So benefit on both sides. That hasn't been done very well in the immuno-oncology field. When I was at Merck, we made a decision to develop Keytruda, which is today a $30 billion a year drug. We made a decision to develop Keytruda with a precision-based hypothesis. And despite the rampant success of that drug in terms of benefiting patients, really nothing has been done in the precision space in immuno-oncology subsequently. And so that's really what we're focused on at Bectas. And the concept here is dynamic versions of precision, because the immune system works in a particular adaptive way. It changes depending on, whether you breathe in, some sort of allergen or not, your immune system is going to respond to that. And so scientifically, when we look at patients' cancers, before we treat them with a drug, and what you can see on the slide here is patients who go on to respond on the bottom row, or those who don't respond in the top row, look no different, or very little difference is seen prior to treatment. However, if you look immediately post-treatment, what you can see is those patients who don't respond on the top row in that lower section and those who do respond are very different now. And so we can identify signals that tell you, as shown by the brown staining on these tumors, these patients will respond. And equally, we can identify the reasons why the other patients will not respond. And this is the concept we describe as dynamic precision. And what it does is allows us to identify the biology behind lack of response in the patients who don't respond. And that means we can find those patients again to treat them with a drug directed against the thing that creates that lack of response. And so we develop a therapy and a companion diagnostic biomarker at the same time. So how do we do this? Well, we take those 80% or 75 to 80% of patients who don't respond. We generate detailed molecular and clinical profiles of those data, of those patients, and then we apply an AI or machine learning-based approach to simplify that down to understand what are the key biological mechanisms that confer resistance and what are the actual proteins driving those mechanisms. And so having done that, we've identified myeloid biology or myeloid-driven resistance as the dominant player in lack of response to existing therapy. And so we're focused on developing drugs there. So what this allows us then to do is to take those patients in the pie on the left-hand side here and identify those that are in that blue portion of the pie. These are the patients who don't respond to existing care because of myeloid biology. And what we can see then is a change in the biomarker as shown in the middle panel. The blue line indicates those patients, the increase in myeloid biology in patients who will not go on to respond to existing care and therefore need something else. By comparison, you can look at the red line. Those patients who do benefit from existing care, you see no increase in this myeloid signal or biomarker. So those where we see the increase, we know that they are not going to respond to existing care unless they get something else. And we can identify those patients in blue who will not respond unless they have the additional benefit of treatment with an antibody that inhibits a protein that we termed AB001. This is a well-known, well-characterized protein. I'm happy to go into that in detail under discussion later. But we know we can identify those patients who will benefit by treatment with an antibody directed against that protein. And we've proven that functionally in preclinical models where we've shown that if we treat animals, as shown on the right-hand side of this slide in the line graph, if we treat animals who have tumors, and they normally grow as shown in the black line in that graph, if we treat them with an antibody against AB001, we can cause over 90% inhibition of the growth of those tumors. And that's just with AB001 alone, which is best-in-class activity from a preclinical standpoint for anything in this space. And mechanistically, that's explained on the left-hand side of the slide, where what normally happens is these myeloid cells suppress the immune system, as shown in the black bar. But if we add Bectas' antibodies, as shown in the purple or red bars, we restore the activity of the immune system. So by restoring the activity of the immune system, we inhibit the tumors. That's exactly the hypothesis we have, and that's what we've now borne out based on the analyses that we've done in a preclinical setting. Yep, thanks. Now, really importantly, if we think about what we're going to do in the clinic, we're going to take AB001 and combine it with existing standard of care, which are T-cell checkpoint inhibitors like Keytruda, like Opdivo, like ipilimumab, which are the well-known drugs that are being used in these spaces. And what we've now also shown is if we take an antibody to AB001, there are certain tumors where it doesn't on its own work. And we would predict that because cancers become dependent upon AB001 when they're treated with existing care. So if we treat those patients with T-cell checkpoint therapies, those only work a little bit, and that's shown in the red bar by comparison to the black. So in this particular graph, the black line are the tumors growing without treatment. The red line are the tumors growing with treatment with existing care. But when we combine existing care with an antibody to AB001, we see the blue line. That's 100% cures, albeit it's in mice, but it's 100% complete regressions in this model, which again is really robust validation of the hypothesis that combination of AB001 with existing care can lead to complete cures in those patients who normally would not respond to the existing care. So where are we with this program now? We've generated a fully human antibody that is primed and ready for movement into clinical development. In fact, we are currently manufacturing that antibody at scale to do all of the studies that are required for an IND, and we hope to file that IND at the end of this year, the end of 2024, and move into clinical trials in that timeframe very shortly thereafter. Pardon? It's an investigational new drug application. So that's the regulatory filing with the FDA that allows us to move into human patients for treatment. So we hope to be in clinical trials at the end of 2024 into early 2025, and the timeline then to demonstrate proof of concept for this as in fact working and demonstrating the value that we're looking for in patients is in 2026, which is a fairly rapid timeline enabled by that precision-based approach. And that's the other piece that's illustrated on the bottom of this slide is we're developing a patient enrichment biomarker to allow us to identify the patients who will benefit from AB001 antibody. So the antibody is positioned to move into the clinic, the biomarker is on the same timeline, and we will have both a biomarker and an antibody ready to use in patients by the end of this year. So what does that mean overall? Well, go back to that original pie chart. We said about 20% of patients respond to existing standard of care, which is shown in the red on the pie chart here. 80% of patients don't respond. We can use our biomarker to identify all of those patients who don't respond, where that lack of response is due to AB001. That's the companion diagnostic biomarker. And that allows us then to treat those patients specifically with the antibody directed against AB001. So we bring all of the benefit to those patients specifically. That increases the likelihood we'll be successful clinically, and it decreases toxicity to the patients. So how many patients is that? Of course, that's an important point. Are we benefiting enough patients? Is there enough market here to make this worthwhile? Well, the reality is precision-based approaches allow us to treat across multiple different cancer types. So we're not just treating patients who have non-small cell lung cancer or just patients who have breast cancer. We can treat any patient in any cancer type that's positive for the biomarker. And that creates a very large opportunity around AB001. Specifically, the biomarker identifies about 200,000 patients per year in the US alone, likely to be dependent upon and benefit from AB001. So that's a multi-billion-dollar market if you think about it from that standpoint, but 200,000 or more patients per year. In addition to AB001, at Bectas, we've identified two other important drivers that account for less patients specifically than AB001, maybe about 100,000 and about 75,000 patients each. But now we're able to address the vast majority of those 400,000 patients who do not respond to existing care. That's half of the patients who don't respond to the best care available to cancer patients today. And we can address almost all of those patients. So in summary then, I think what we built with Bectas already is a highly experienced team with deep scientific and operational experience. We're founding our insights on our programs on insights directly from human patients by studying cancer patients and why they don't respond. So we know if we can address those reasons, these are going to work when we bring them into development. That's led us to myeloid biology, for which we have 3 programs, the most advanced of which is currently in manufacturing, ready to move into clinical trials at the end of the year. And we have a very specific biomarker to identify patients who will benefit from that drug. And that is really going to accelerate our clinical development. So we're on track for the IND at the end of this year and clinical proof of concept in 2026, which is a very rapid and capital efficient demonstration of clinical proof of concept. So where are we from a capital raise standpoint? All of the initial work was done using a SAFE or a convertible note type approach, a SAFE approach, which has allowed us to found the company, generate and initiate these programs and bring them this far forward. We've done that in the course of about 15 months, which is about half the time it would normally take. So highly efficient from a capital standpoint, highly efficient from a timeline standpoint, and we have a biomarker and a drug in place. We're now raising a SAFE extension of about $2.5 million, which we're trying to close in January. We've got significant interest in that. We're certainly looking for additional investment in there. There's some opportunity to take oversubscription in that, but not by very much. And so that will allow us to hire the team, maintain the accelerated progress that we've already had with the Bectas leading program, and allow us the time to build the Series A syndicate. And we've already have some interest in there. We're looking at talking about developing lead investors in the Series A syndicate. And so the plan then is to close on that Series A in 2024 as well. So investors in this round of the SAFE will convert fairly quickly into that Series A. And that Series A should fund us through clinical proof of concept and a profile for a biotech company, which is fully consistent with a public offering if the markets were appropriate, but certainly with clinical proof of concept. In the myeloid biology field, there's a significant amount of interest from pharma in that sort of program. And in fact, we have a lot of inbound business development interest. And later this afternoon, I'm actually starting on the series of meetings that I have this week to talk to pharma about business development around the later programs in Bectas. So I think we're in pretty good shape there. Yep. So I'm just going to summarize. Pardon me? Yeah. So currently we're looking at a 15% discount. And a pre-money value cap of 15 million.
Sorry,
15% discount and pre-money value cap of 15 million. Happy to talk about that in more detail as well. Yeah. That's just my contact information since I think the last time when I met with with Marty and the team and the group here. I didn't have my contact information as clear, so I thought I would put that up on the last slide and leave it for anybody who wants to screenshot it. More than happy to have additional conversation about this afterwards, either today or later, and happy to answer questions at this point.
Any other questions? Yes, can you talk to us about how much you've raised so far? Is there an insurance code for what you're trying to do?
Is there no? So this is drug discovery and drug development. So it doesn't fall under that sort of same direct care at this point in time. It's not a commercialized drug. So we're in the biotech business building the program through, we'll bring it into clinical trials later this year. And then there'll be a timeline to FDA approval of that, at which point there would be a billing associated.
Part of what you do is you're discovering a biomarker, is that correct?
Yes. Yep. Yeah. So there is a regulatory process for that as well. That's also later. So as part of the development, we can develop the biomarker and the drug in parallel, hand in hand, but will still be under the guise of a clinical trial.
Yes, sir. Yes, great job. Thank
Okay.
you so much.
All right. Thank you.